Ina from St. Jude Children's Research Hospital, and together with Judy, we are chairing today's session. I think pause was enough. I hope you could feel your coffee or water, and you're back to hear about the tools that are pushing our field forward. We are in this session 4 about innovative methods and technologies. And we are going to hear about tool compounds, NMR structural studies, novel biosensors for deorphanization. And lastly, our field is also introducing various computational pipelines. This one, which we are going to hear from Marta, is about variant prediction. And I'm just realizing that spans like a beautiful range of disciplines, from medicinal chemistry, structural biology, cell bio, computation. So we are really going strong on all fronts. I think that's beautiful. Future is bright for sure. And first one will be Lars Jürgenliemke. Lars will tell us about Glue-tiful inhibitors. Please take it away.
Thank you very much, Katharina. I hope you can hear me. So first of all, thank you very much to the organizers for giving me the opportunity to actually convince you of our story of 2 inhibitors that are Glue-tiful, meaning they enhance the proximity within the subunits of the heterotrimeric G protein. So we wanted to talk about innovative methods and technologies. And many times the basis of these are pharmacological tool compounds that are very handy to investigate GPCRs and their downstream signaling. So yesterday we already heard a lot about tools and methods using labeled proteins, overexpression, and the famous knockout cell lines from ASCA and also Carsten's lab. But when we ask ourselves whether results from those experiments are transferable to unmodified primary cells, organoids, or even model organisms, pharmacological tool compounds become especially valuable. The 2 natural products, FR and YM, are actually prime examples of those pharmacological tool compounds. To be more specific, they are inhibitors. They both derive from bacteria, in case of FR, synergistically living in this nicely looking plant, Adisia crenata, and in chromobacteria subspecies in case of YM that live in the soil. They are designed with a perfection that I guess only evolution can achieve to fit into the intermolecular cleft between the RAS and the alpha helical domain of the GDP-bound GQ family proteins. And thereby, they are able to inhibit the nucleotide exchange, which is why they're also called GDIs, which is short for guanine nucleotide dissociation inhibitors. And this makes them able to potently inhibit G protein signal transduction with unrivaled subfamily specificity. This mode of action is already supported by an X-ray crystal structure of YM in complex with heterotrimeric GQ that was obtained by the Ito group in 2010 and was assumed to be similar for FR based on MD simulations and mutagenesis studies. Now, if we take a look at our potential toolbox of G protein inhibitors for all the subfamilies, which would, if complete, enable all of you to take a more holistic approach to the study of GPCR signal transduction, we do see that only GQ and GI family proteins can be inhibited by their respective inhibitors, FR and YM, or pertussis toxin. And this is not due to the lack of trying to find new inhibitors, rather than difficulties on the journey. So there have been some promising compounds that have been identified, but they do all come along with significant hurdles like poor cell penetration, cytotoxicity, or just lack of complete G protein inhibition. And just to give you a prominent example you might have already heard of, the nucleotide state-specific binder GD20 that was published by the group of Kevin Schuckert in 2022 is a molecule that selectively binds GDP-bound G-alpha-S. And thereby it acts similar to FR and YM as an inhibitor of nucleotide dissociation. But still, it is unable to globally inhibit G-alpha-S signal transduction. So the question here is, what makes FR and YM so successful? And when we look at the already published structure, this one does imply but not resolve a contribution of the G-beta gamma dimer which is a G-alpha inhibitor itself. And our aim in this project was now to obtain a better understanding of these inhibitors in the context of the whole heterotrimeric G protein, to then probably one day be able to expand the G protein inhibitor toolbox based on this knowledge. And therefore, we obtained two new high-resolution X-ray crystal structures for both FR and YM in complex with heterotrimeric G11. This was done in collaboration with the group of Gebhard Schädler at Paul Scherrer Institute in Switzerland. And the structures show indeed that FR, just like YM, binds into this intermolecular cleft between the RAS and the alpha helical domain, and thereby locks the GDP inside of the protein. So we already have this published as a preprint on bioRxiv if you're interested in the overall structure, because moving on from now, I want to focus on one specific interaction, which is a residue in G-beta, arginine 96. And this residue was neither observed nor predicted before to interact with the inhibitors. And it could, at least that's what we thought, very well be part of the hidden intricacies that make FR and YM so incredibly well suited to completely blunt GQ signaling. And the aim of our group here was then to unravel the biological effect of these interactions, which we did by site-directed mutagenesis of the arginine 96 residue, and then the investigation of inhibitor properties in cellular assays. And we focused first on a very crucial inhibitor property, which is the potency. And we investigated this via the free G-beta gamma GRK breadth sensor provided by Kirill Martimianov's group, which consists of a Venus-labeled G-beta gamma subunit and a nanoluciferase-labeled mass GRK3CT, which is a G-beta gamma effector mimic. And upon activation and dissociation of the G protein, the interaction between beta gamma and mass GRK leads to a high breadth ratio that can be prohibited by the addition of the inhibitors. And then we recorded potency curves for both FR and YM, first in heterotrimeric G11 with G-beta wild type, and then we mutated the arginine 96 residue to alanine and to leucine. And what you see here is that the interaction with this residue in G-beta has no impact on the inhibitor's potency. That means the residue is not essential for FR and YM's mode of action. So great, we thought, what does this residue do then? And we found the answer to this in data that was also provided by Gebhard Schädler's group from Switzerland, which is a thermostability assay, where we measured temperature-dependent protein unfolding as a measurement of protein stability. And you do see in gray the apostate of the heterotrimeric G11, and in magenta and blue, the state where FR and YM are present. And you do see that upon FR and YM addition, the denaturation, the curve shifts towards denaturation at higher temperatures. Additionally, when we form the first derivative of this data, you do see that there's a plateau in the apostate, indicating two distinct unfolding events. First, the G-alpha subunit, and second, G-beta gamma. Now, when we add FR and YM, you do see that only one unfolding event is detectable in addition to the already mentioned temperature shift. So this indicates that the inhibitors not only inhibit GDP dissociation by their direct interaction with G-alpha, but also act as something like bioglues that enhance the protein-protein interaction within the G-alpha and G-beta gamma subunit, and thereby probably also utilize the silencing properties that the G-beta gamma dimer has on G-alpha. Now, since this was done in purified proteins, we wanted to take this back into experiments in living cells. And we utilized here the same breadth sensor as before, which this time only in the basal state, meaning without activating the G protein by a receptor, detects a spontaneous dissociation of the inactive G protein. And if we add the inhibitors into this equilibrium, and if our stabilizing effects would matter, the basal breadth ratio should decrease because of the enhanced protein-protein interaction between the alpha and beta gamma subunit. And this is indeed exactly what you can see here. So upon inhibitor addition, you can observe that the curves for FR and also YM decrease. This indicates that the observed heterotrimer stabilization does also occur in living cells. But we still wanted to further confirm that what we see in this assay actually is the stabilization of the heterotrimer. And therefore, we leveraged a disease-causing mutation, which coincidentally also occurs in the arginine 96 position in G-beta, where arginine 96 is mutated to leucine. This mutation is reported to increase G-alpha and G-beta gamma binding, and it thereby acts as a stabilizer of the heterotrimeric G protein itself. Therefore, the addition of the inhibitors should have no effect on our bread assay. And as you can see here, the effect of FR and YM is at least, I would say, severely compromised by this mutation. Now, in the next step, we wanted to trace the effect back to the specific residue in G-beta, and we mutated it to alanine. Again, you see in thermostability assays, the mutation has no impact on the apostate, so without the inhibitors present. But when we add FR and YM, you see that for the arginine 96A mutation, the stabilizing effect of the inhibitors is less pronounced on the mutant. And similar results can be observed in the bread assay, which, when we compare it to the wild-type bread assay, you do see that the effect for the mutant is much smaller, indicating that this residue in specific, but also G-beta in general, play a role in the stabilizing of the heterotrimer. And I hope that with this, I was able to convince you that our data indeed shows this. And in the last step, I want to go back and compare FR and YM to the other GDI, GD20. Because this turns out to potentially be the crucial difference between these compounds. While FR and YM inhibit nucleotide exchange while tolerating the inhibitor's occlusion of G-beta gamma, while GD20 excludes G-beta gamma in its action. And we think that probably this aspect is necessary to inhibit the entire heterotrimer and could therefore enable the development of novel G protein inhibitors that could fill the current gap in the box of our pharmacological tool compounds. And with this, I would like to thank all the people in our working group and of course our close collaboration partners from the Institute of Pharmaceutical Biology who never get tired of providing FR to us. And of course our close collaboration partners in this project from Paul Scherer Institute and also University of Marburg. And of course you for your attention.
Thank you very much for this engaging talk. I see a raised hand. Marcel, ask your question, please.
Yeah, thanks for this very Glue-tiful talk. When you talk about molecular glues or you introduce the concept of glues, it's not far away to protein degraders and even proteins. Do you think that FR and YM peptides are suitable for designing proteins? And if yes, is anchor 1 or anchor 2 the better choice?
I mean, that's a good question. I think in principle, so they have really high affinity binding to the G protein, to the G alpha subunit. So why should they not be able to be developed as protein? I think it's called warheads. So anchor 2 is really deep inside. Anchor 1 is really deep inside of the G alpha subunit. So then it would probably be anchor 1, but also the interaction with the G beta gamma subunit that we mentioned here is mediated by anchor 1. So what seems to happen is only slight changes in the molecule lead to a great loss of affinity to the target protein. This is something that could be problematic when you want to design projects with FR and YM.
Thanks.
So thank you again, Lars, for this talk. I don't see any more raised hands and no questions in the chat. As always, there's going to be a lunch session right after this session where you can ask additional questions. I would like to introduce the next speaker. We already see the video. Libin is going to talk about QNMR, assisted structural elucidation of an intermediate GPCR-G protein complex.
Can you guys see me?
Yes, we see you,
Okay.
we hear you, and we also see your full screen slide.
Oh,
Thank you.
sounds great. So thank you very much for giving this opportunity to talk about research happening in my lab. So today I'm going to talk about the conformational dynamic and function of LAD medium GPCR-G protein complex. So I'm currently an associate professor in the Department of Molecular Biosciences at the University of South Florida at the Murphy Cancer Center. So as we know, mutation causes diseases, right? So if the mutation happens in the functional domain, such as in GPCR, in the ligand binding domain and the G protein binding domain, that's easy to understand. But we do have a lot of mutation. Actually, there are a lot happening in the functional domains. They do cause diseases. That's why I want to introduce you concept between the conformation and the diseases. So what is the molecular mechanism behind that? I personally always think because when mutation happened, the conformation equivalent of the receptor changed. And of course, the allosteric signal pathway changed. That caused diseases because as we think about different conformation, maybe interact with different G proteins or maybe later on GRK or beta arrestin, right? So from this perspective, if we can understand the function of each conformation states, can help us to design drug more purposefully. So to understand the conformations, actually in last decades, I think the structural biology tool is very, how to say, a dramatically advanced field, especially X-ray and color-EM. As all you know, these things in the past year, especially the color-EM from the 2013 and 2017, the first GPCR-G protein complex resolved. So far, we have resolved about 1,000 G protein, GPCR-G protein complex structures. Okay, but if you look at those structures, and if you sort them, you will find actually maybe the 30% or 50% of the structures in active state, and all the structure resolved for the complex G protein, GPCR-G protein is represent the fully activated complex structure. This may be let us think about maybe GPCR still adopt the two-state model, but as we know now very clear from different kind of the sources, we know GPCR activation process is a multi-state transition process involved from the complex perspective, at least involve 3 types of the complex, such as pre-coupled complex with the G protein, and the partially activated complex structures, and then the fully active complex structure. But we don't have these 2 complex structures in the database, to be honest, right? Because they are very dynamic and it's very difficult to catch up. That's why I come up with our project. So if we want to understand their functions, so first we need to catch them, right? Or then isolate them, then study them, and understand the function of each conformation states. There's a couple of tools now in the field, such as some people already talk about DO experience or the fluid experience. I want to introduce you the NMR experience. So as you know, maybe you touch a lot about a conventional NMR, such as proton, carbon, nitrogen. But here, we are talking about a fluid NMR. So fluid nuclei is a very sensitive microenvironment change. That means any change of the conformation could reflect on the spectrum of different peaks as shown here. So each of the peaks on the NMR spectrum represent a different conformation states, such as you have the conformation state 1, conformation state 2, conformation state 3, and we also can calculate how fast we use the decay experience, how fast from S1 to S2, how fast from S2 to S3, because this is very important. Actually, is missing in our current drug discovery system, such as if you want to profile action of the player's basketball, you want to put a ball into the basket, right? First, we need a right conformation, right pause in order the ball go to the basket. If the pause is not correct, you cannot use a play circle to play basketball. That doesn't work, right? So second, how fast I was moving on your hand determine how many ball you can put into the basket 8 minutes, right? So these two perspective, one we call conformation, another called the dynamic, are very important, but it's missing in current drug discovery system, right? So with this in mind, actually my lab tried to delineate different conformation states of the receptor, and understand the functions of each conformation states, right? Maybe different conformation maybe interact with different G protein, GRK, or beta lactate, etc. So this is from the AMR spectrum we acquired. This is a example because we have a label, so I label as we have a fluorine tag, so I label the white type of star. So you see the bunch of the peaks, right? So each of the peak represents a different conformation states in this case, because there's only one probe in this system. We label on the TM6. Then at the beginning, we talk about the mutation, right? So you can see it's very interesting here. If you make a mutation, you see this is a example. We didn't add any ligands into the system. You can clearly see when the mutation happened, the profile of the conformation equivalent changed, each of them. Especially as R2 line 1, you can clearly see the S4 state accumulated. So we define the S5 state fully active state using the with addition of the 4 agonists and plus the G alpha beta gamma, right? So from this perspective, actually now in this mutation, we can particularly accumulate the intermediate state S4. Then we can start the S4's function here. This paper actually, we didn't publish the complex structure, but we did publish a different state structure in the lecture communication last year. So in this case, actually, because we can particularly accumulate S4 state without interference from the S5 state. S5 state is the fully active state. Then we can start a couple of questions here. Whether S4 state itself can interact with the G alpha beta gamma. Second, whether S4 state can regulate nucleotide change independently without transition to S5 state. Because in this mutation, actually, we block the S4 state transition to the fully active state S5, right? So the next question, how the G protein engages the S4 state? All this question we can answer. Because it is the intermediate state is very dynamic, so I collaborate with the event transient from the UCSF, and it took us about 1.5 years. Eventually, we got the structure resolved here. So this is a 2D classification, and this is the model we established so far. And actually, we put the paper into the BioQ already, but we still do some refining stuff now. But this is NMR spectrum. This is the convoluted NMR spectrum. So we're using the crystalline structure of the fully activated A2A with G protein structure as a reference. And this is the NMR can clearly see in our mutation, you add the G alpha beta gamma, you add the G alpha beta gamma with midi G, as the conformation cannot shift to the S5 state, right? That means the structure will solve the intermediate complex structures. Of course, we also using the, because in this structure, we still don't have the AHD domain. This could be a next plan we are trying to do. But we also use the computational simulation. We look at the what's the difference between of the G protein, especially the C alpha 5 domain and the H1 domain. You can clearly see in the fully inactive structure of G protein, there are two interactions. So we call the CH3 pi interaction and H bond is fully closely formed. In the fully activated structure of G protein, they are broken. But in the intermediate complex, there still are weak H bond between the CL5 and H1. Okay, and this actually represents this structure here. Okay. So we also want to know the function, because in this case, we can avoid the interference from the S5 state or S5 complex. So we want to know S4 state, how S4 state regulates G protein, if S4 state can regulate G protein, right? So this is, we did the first, did the GTP hydrolysis experiments. So this is the Y type. You can clearly see the Y type can hydrolyze the GTP extremely fast. And this is R2-1 mutation. Whatever you add as a full agonist or partial or the inverse agonist, their hydrolysis ability is very limited. They cannot go to the depot. Okay. At the same time, we look at the nucleotide change. We use the body GDP. Of course, we have the other body GTP as well. I didn't show that here. So we can clearly see this is S5 stem. You can see associate and dissociate at good balance. But the S4 state regulate the G protein, with the GDP, you can see associate, dissociate, and you see second association very slow. So that means there's a two binding site in the G protein. From this perspective, we try to figure out what's going on here. But this also indicates the AHD domain in this as a four-state regular G protein is not fully opened. Okay, so just summarize, actually, from this study, we saw the first intermediate GPCR-G protein complex. Second, we provide a direct evidence that a nucleotide change is initiated at the intermediate complex stage instead of the end state GPCR-G protein complex stage. So third, we also established a strategy of using following AMR to guide the structure and the function study of the intermediate GPCR-G protein complex. The fourth one, so this could be in the future, allow us to integrate a confirmation-based drug development dimension into current drug discovery system. Here, as we nowadays, we learn a lot about the structure-based drug discovery and the cell-based drug discovery. In the hope someday we can integrate the confirmation-based drug discovery into the system. Because I always advocate the signaling bias from the confirmation bias. Why the different G protein, same G protein can go to different G protein signaling or beta arrestin signaling because they use different confirmation states. The most important thing now, we need to delineate different confirmation states, understand the function of each confirmation states, that we can guide us to design drugs purposefully. Okay. So at the end, I want to thank you for all my collaborators and especially my students, Xu Wang. He's involved a lot for this project. I have a lot, in the last 5 years, I have a lot of students at AD McFarman. He's working on another project of interesting dialogue. Fragile GPCR is not talking here. So I also want to thank you for all my funding from Enlarge, NS, and especially I also serve as consulting faculty now for the Eli company. At the end, my lab is continuing to recruit the postdoctoral fellows, and my department, actually, I'm serving the associate chair. We also recruit a tenure assistant professor in the direction of the molecular biophysics. If you are interested, I think we are going to announce it very soon next week. If you're interested, you can contact me or you can submit your application as well. At the end, thank you very much, all of you, and I'm happy to take any questions you have. Thank you.
Thank you, Libin. That was a beautiful story showcasing how you can study certain conformation by creating a mutant and trapping it in the certain conformational state. So I saw Judith had a hand before. Do you have a question, or was just that to make sure we are on the time?
Oh, I do have a question. So right now
Perfect.
you're looking mostly at the conformation of helix 6, right?
Yes, so yes, we use a helix 6 as the indicator.
Could you also look at conformation, like with this method, look at the conformational states of the rest of the receptor and then combine the information maybe?
Yeah, that's very good ideas. Yeah, of course, but yeah, we do have different label site. Actually, we publish a paper. So from a lot perspective, so when you profile the conformation states, you want to find assign the most or represent the larger scale of the conformation states, right? So from this perspective, you don't need too many sites, to be honest, you know, right? So in principle, each site represents contains all the conformation states. The reason you cannot see that is just because of resolution issue. So such as in AMR, we clearly can do the 5 states. I always think the receptor, just like us, they just stay in the microenvironment. We play basketball, play soccer, use different conformations. That's different functions. So receptor also use a different conformation, doing different functions. The important thing is how we delineate them and then study their functions. Yeah.
Thank you.
Yeah.
Thank you. And beyond the time to present our next speaker, that's Luca Franchini. Luca will tell us about his novel assay for deorphanization. I think that's very relevant. We still have more than 100 GPCRs which don't have a ligand. So let's see if Luca's assay can help deorphanizing something.
Do you see my screen?
Yes.
Is the presentation one, right? Okay. Yeah, thank you very much for inviting me for this talk about innovative method and technology session of the GPCR Symposium. I'm glad to share with you some insights about my main postdoc project, which is the GZESTY signal transduction assay, GZESTY, for GPCR deorphanization. Our group focuses on orphan GPCRs expressed in the brain, which lack information about ligands and signaling components. So our goal is to identify these receptor features. From a recent publication of our group, it appears that most of the orphan GPCRs in the brain activate inhibitory G-alpha-IO proteins, which downregulate adenylyl cyclase and reduce CMP level. However, if you are working with G-coupled receptor, you know how hard it is to deal with them. In fact, these receptors are very selective for the type of G-alpha protein subtype they couple to, and some of them don't even couple to beta-arrestin, like GABA-B. So this research strategy on top of which high-throughput screening assays are based for detecting GPCR activation is fundamental in the pipelines for identification of ligand receptor pairs, especially for this type of GPCRs. So we came up with the GZESTY signal transduction assay, GZESTY, which is a cell-based assay in which we transfect HEC293 T cells with the assay components, such as the G-coupled receptor of interest, or orphan GPCR, the GS-GZ chimera, which bears the C-terminus of GZ, responsible for decoupling to the receptor, but the core is of GS, responsible for the function, stimulating adenylyl cyclase. We also co-transfect the Glue sensor, which is a splitted 5-luciferase branded from Promega, which is able to bind CMP, and upon this event falls in an active conformation of the enzyme, converting the luciferin into oxyluciferin and emitting light. This light emitted is proportional to the CMP produced. We also co-transfect in the assay PTX, pertussis toxin, which adiperibosylates endogenous G-alpha-IO proteins in the cell model and impedes that this G-alpha protein can couple to our receptor of interest. In this way, when we activate a G-coupled receptor through GZESTY with the correct agonist, what happens is that we have activation intracellular of the chimera GS-GZ, which stimulate adenylyl cyclase. We produce then CMP, which is accumulated over time in the cells. And this CMP is detected by the Glue sensor, emitting proportionally light, which is then detected by our plate reader. So we hope that in the future, this assay can allow a easy ligand receptor identification, especially for G-coupled receptors, but can also be used for high-throughput screening platforms and for GPCR deorphanization pipelines. So at the beginning of this project, I started setting up the assay, and I chose 4 different G-coupled receptors, 3 of class A, D2 receptor, dopamine D2 receptor, adrenergic 2A receptor, and mo-opioid receptor, 1 of class C, like GABA-B. And I tested them with 3 already available GS-based chimeras, GS-GO, GS-GI, and GS-GZ. And I measured receptor activation as a fold change. As you can see in darker color, GS-GZ was the chimera that for all the receptors was mostly fitting and allowing to detect the receptor activation. So I continued maintaining GS-GZ in the system, and we thought to combine it with pertussis toxin, given the fact that this enzyme does not affect GS-GZ. So it is able to impede the endogenous GIOs, but our chimera is intact. So whenever I co-transfected a very low amount of PTX, you can see that I already maximized the fold change detected. And also when I titrated the amount of receptor plasmid to be transfected, what I noticed is that the highest the amount of receptor transfected, the higher the fold change. So I came up with an optimal ratio for this multiple plasmid transfection, in which you can see in the checked pattern, the lowest amount of chimera and the lowest amount of PTX combined with the highest amount of receptor transfected provide the highest fold change detected. Then I started testing different experimental conditions that could affect our readout. For example, our temperature, working temperature. I noticed that 28 Celsius was the best working temperature for this assay. I also tested the possibility that some media components could affect the assay, and I compared them also with a serum starvation that is usually a common procedure for GPCR assays. And then I verified the possibility to use coated plates to perform the assay. I also then checked the possibility to co-apply phosphodiesterase inhibitor, because as you can imagine, we are basing all this assay, this readout of receptor activation as an accumulation of CMP, stimulating adenylyl cyclase. But if at the same time we block degradation of CMP, we should increase the detection of receptor activation. And indeed, this is what I noticed, and this by co-applying IBMX. So this is also an extra feature that can be added into the system. So we can summarize this first part of the setup saying that there is an optimal ratio of the GZESTY component to be transfected for a maximal full change. There is an optimal temperature for working with it that is 28 Celsius. And there are some extracellular matrix interaction that sometimes GPCR engage and can be relevant for some receptor for their stabilization of the plasma membrane, for their signaling. And you can use this, say, on coated plates in this scenario. You can also add the IBMX into the system to increase the full change detected. However, some of these situations should be revised depending on the GPCR that we're interested in, that are unique and peculiar for each feature. Then we wondered if this assay could be suitable for high throughput screening. That is what we want to use the GZESTY for. So I measured the Z factor, which is an adimensional parameter, providing a reliability of the positive hits of an assay. So basically, it tells us how much the positive hits are specific and not random. The Z factor should range between 0.5 and 1 for a good high throughput screening. And when I tested it for the GZESTY assay, it was 0.78. So it matched this high throughput screening definition. So what can we use the GZESTY for? What are the applications? First of all, we wanted to develop an easy cell delivery method for the GZESTY component. And for this purpose, we collaborated with John Luec from our department, who works mostly on molecular biology approaches. And they recently set up a novel cloning technology through which we can assemble components of interest with specific promoters into a single plasmid. This allowed us to basically maintain this optimal ratio that I previously showed you for the transient transfection that are relevant for detecting receptor activation in this type of assay. So we chose strong promoters for the glow sensor and the receptor, but we chose a very weak promoter for Chimera and PTX. And then we obtained through their technology a single plasmid that could be transfected with multiple components integrated. And just to show you a comparison of the approach with a single plasmid bearing the 4 components or the multi-plasmid transfection, basically I didn't see great differences. So this allows us also to have a faster transfection without having to calculate complicated ratios between plasmid, etc., etc. The plasmid are also suitable for baculovirus production, so you can also use them to infect cell lines that are harder to transfect with common reagents. So I used the 3-in-1 plasmid and I tested a battery of geocoupled receptor at this point with the respective known agonist, and I noticed activation of over 90% of them. Given the fact that we are measuring receptor activation as an increase in CMP levels, I also tested the GS-coupled receptors with the same approach, and I noticed activation of them. So overall, we can say that expanding also on the GS-coupled receptor 1, GZESTY allow you to detect over 86% of the whole GPCR. Then I tested the GZESTY assay for detecting a specific pharmacological profile of different agonists at mucoid receptor. And as you can see, damgo and beta-endorphin, which are known full agonists at mucoid receptor, provided very similar maximal efficacy, while morphine, that is a partial agonist, much lower, confirming the results in literature of their pharmacological profile. I also tested the activation of endogenous GPCRs, such as PAR1 and PAR2, that are expressed in X cells. And I compared them for their activation with a 2-in-1 plasmid that does not encode the chimera. This is why, because PAR1 and PAR2 do not couple to GS, but only to GI, GQ, and G1213. So only when the chimera is expressed, I can see activation of these receptors. Then we come to the final, most important for us application, that is the deorphanization pipelines. So historically, GPCRs have been deorphanized by application of tissue extracts with different origin, vegetal, animal, etc. And this as a source of ligand that could activate or bind to the receptor. So we thought, let's test a battery of geocoupled receptors that are expressed in the brain with a mouse brain extract and see if we can detect activation through the GZESTY assay. So while our battery of G-coupled receptor that I regularly used is all expressed in the brain, we know it, and they showed actually activation with the mouse brain extract. What was more interesting for us is that two orphan GPCRs that are also expressed in the brain and that are JO coupled, GPR-176 and GPR-37, also showed consistent and strong activation in the assaY, suggesting that the ligand of these receptors is in the brain. In particular, GPR-37 has some literature report, but the results are kind of conflicting between the confirmation of what is really the ligand for this receptor, while for GPR-176, there are really no information about a ligand. But it is known to be a G-coupled receptor expressed in the brain coupling to GZ. So with this, I will take any question, and I thank all the lab component, the collaborators, the department, and you for the attention.
Thank you very much for this talk. I see nothing in the chat yet, and I also don't see anybody raising their hand. So maybe I can ask you a question. So how would you proceed now with the last part that you showed with the brain extract? Would you start to fractionize it and test it and start to figure out what compound exactly is the one activating the receptor?
Yeah, so we are now working on pipelines for fractionating the mouse brain extract and trying to verify what could be the ligand. There are some hints in literature of the nature of the ligand for this receptor, just for the nature itself. So if it is a protein, a peptide, or a small molecule. But definitely we have to find like a pipeline that could work and where we could actually apply the GZESTY to verify then the presence of the ligand in that fraction. So yeah, I already have set up like a sort of general procedure to work with, and maybe with some collaborators of the department that perform HPLC, for example, or that are more biochemically focused. I can improve some steps of it. So just to make sure that I'm really able then to find a possible ligand for it. Yeah.
There's also one question in the chat. I think we have time for that. From side, what impact does the expression of the vector have on endogenous GZ?
Of the vector, what do you mean?
Maybe, Side, if you're here, you could shortly unmute yourself.
Let me see the chart also. I didn't see that. So there is no endogenous GSGZ because it's a chimera, so it doesn't exist in nature by itself. Hex cells that we use display GIOs, especially GI, but in our hands, we test them for example, for GO, and it doesn't seem that there is GO, and it doesn't even seem that there is much GZ, to be honest. I have some extra data that are in the paper that is on bioRxiv if you want to check it out. And I used also oxytocin, that is a toxin that blocks also GZ. So it blocks basically the wild type GZ, but still not the chimera. And so when I have oxytocin transfected instead of PTX, I still see the same fold change with PTX. So it doesn't seem that there is really a great contribution of GZ, if it could be expressed or not, regardless in our cells in the amount, because I have the data about it. So yeah, endogenous GIO are basically blocked. We continue with PTX just because we noticed that GZ was not really affecting anything. So we just preserve PTX for common sense, but one could use oxytocin as well.
Perfect. Thank you again for your talk and for answering the questions. Our next speaker is Marta. I don't see her right now. If you could.
Yeah,
there
hello.
she is. Hi.
Okay.
Your
Okay.
talk is called GPCR-specific pipeline for variant impact predictions. And I'm excited to hear you talk. Take it away, please.
Okay. So I hope you see correctly the screen and full screen. Okay.
Yes.
So hi everyone, I'm Marta Lopez-Balastegui from Jana Selen's lab at Barcelona. I will like to thank again the organizers for this great congress and also for selecting my talk. I am very happy to share with you how we are trying to develop this GPCR-specific pipeline for variant impact predictions. I would like to emphasize that this is an ongoing work, but I will try to convince you that our approach is a good approach. However, if you have any ideas or suggestions, you would be very welcome to come to the poster session afterwards to share them with us. So with that, I will start talking a little bit about the knowledge gap that there is currently in the impact of genetic variance predictions. Then the performance of the existing predictive algorithms. And finally, how we are trying to build a GPCR-specific tool in order to predict the pathogenicity of variants, particularly affecting GPCRs. So regarding this knowledge gap, there are more than 4 million observed missense variants in humans. However, only an estimated 2% of them have been clinically classified as pathogenic or benign. Since the early 2000s, several algorithms try to predict the effects of these untagged missense variants. In general, predictive algorithms work like this. You introduce your missense variants of interest, and you get an output that it's a number or a probability. That with that, you can build your classification. So you can tag your variant as predicted to be benign, ambiguous, or pathogenic. So how the existing predictive algorithms perform at this task? In here in this plot, you can see in the y-axis a collection of predictive algorithms that try to predict the pathogenicity of missense variants, and in the x-axis, their performance. So what we can see here is that alpha missense is the best performing tool. However, if we compare this algorithm with the rest of them, we see that we have a modest improvement. We wanted to comment on that because alpha missense was published last year, and several of the authors of alpha missense are also authors of AlphaFold. So after it was published, there was like an expectation that alpha missense was going to be a game changer in the field, as AlphaFold was in the prediction of the structure of proteins. So what we see here is that it's indeed a really good algorithm, but it's not having an impact as AlphaFold had in its field. In addition to that, alpha missense benchmarks also revealed a high number of false positives, as stated in this paper here published this year. And we also, as you will see in a couple of slides, face this issue. What we want to say with that is that alpha missense is really a good tool. However, there is still room for improvement. So our strategy is to build on top of alpha missense by adding GPCR-specific features to try to reduce the number of false positives, specifically working with missense variants affecting GPCRs. So how we are doing this? Our idea is to analyze GPCR-specific features, as for example, check the allosteric network presence. So check if the residue that has the variant is present or is highly important in the allosteric network of the receptor. Then also we check if there is a significant connectivity variation of the position of the residue, having the original residue or the alternative one. Also, if there are interactions with the ligand, and the location and orientation. This is an important feature because although alpha missense shares the same architecture as AlphaFold, it means that it incorporates the structural context, so it takes into account where is located the variant in the receptor. It lacks the information of the surrounding of the protein that you are interested in. So, for example, it does not have into account the membrane surrounding of the transmembrane residues of GPCRs, and we are also adding this information. We also check the change of charge, if the substitution supposes an helix breaker, and the value of the substitution in a matrix that was trained with GPCRs. So in addition to that, these top 4 features that you see in orange are also adding information on top of alpha missense because they are extracted from molecular dynamics. So they also incorporate this dynamic information that alpha missense is just using this static structural information. So our plan is to introduce the missense variants affecting the GPCR that we are interested in, check the prediction of alpha missense, and before having our final classification, we check the cases in which we have a positive. That is the cases in which alpha missense predicts the variant to be pathogenic. So the idea is to try to identify false positives with our GPCR-specific feature analysis and try to rescue them and correct them. How we are refining alpha missense. So we are using this a result from this paper that was published last year. They did a collection of very interesting analysis from which we used this phenotypic score that they computed for 60 different missense variants affecting the GLP-1 receptor. So based on the analysis of the surface expression and the analysis of these 4 different signaling pathways, they were able to assess a phenotypic score so as an experimental value of the impact of each of the variants of the dataset, so we can classify them as benign, ambiguous, or pathogenic. So with this data, what we can do is that we can compare the predictions of alpha missense with the phenotypic score. In this plot, you can see in the y-axis the predictions of alpha missense, and in the color scale, the phenotypic experimental values. With that, we can set different thresholds. And what we can see in this plot is that for these low values of alpha missense and medium values, we have darker colors, meaning that we have here like an agreement, a correlation with the experimental values. However, in this top region of high values of alpha missense, so predicted to be pathogenic, we have lighter colors, but we also have darker missense variants in here, meaning that we have false positives. So we plan to check the GPCR-specific features to this group here to try to identify and rescue these false positives. So how we are doing that? We can take a look in detail to these 4 positives that we have detected and start checking the GPCR-specific features. So for example, checking the allosteric network presence, we saw that for all 4 missense variants, there was a low contribution to the allosteric network that mediates the signal from the ligand to the intracellular site. In addition, we can check the location and orientation of all of them. And we saw that 2 of them interacted with the hydrophobic tails of the membrane lipids, and 1 of them interacted with the polar heads of the lipid. And the cool thing about this feature is that we can check in detail if the substituted residue is going to be also comfortable in this particular position, taking into account the GPCR-specific surrounding. Then finally, with the other features, we can also check that these 2 of them were indeed homologous and well-tolerated substitutions. So all in all, with that, what we can do with our GPCR-specific analysis is that we are able to first identify and then rescore them in order to be moved in this scale. And now you can see that the correlation is fitting much better. So we have darker colors for the lower values of alpha missense, and in the top region, we have reduced the number of false positives. So we have here lighter colors. We can see this improvement better with these plots. In here, you can see in the y-axis the alpha missense prediction, and in the x-axis the phenotypic score. And we can see in this top left part the 4 false positive. If we run our GPCR-specific analysis and refinement, we see that indeed we rescue them and we can improve the correlation. So summing up, we have seen that alpha missense shows a good correlation with the phenotypic score, but there is still room for improvement. For example, because alpha missense ignores the membrane environment of GPCRs. Also, that the implementation of GPCR-specific features could be critical for improving the variant impact prediction. We also know that the dataset that we are working with is small, and in this case, currently, it's tailored for the case that we are working with and lacks generalization. However, we are about to access a larger dataset in order to generalize this model. And we also aim to introduce new GPCR-specific features, as for example, if the residue is in contact with G proteins. And finally, thanks to our collaborators in France, in CERN, we will be able also to experimentally check if our predictions are correct or not. So that will be all. Thank you all for the attention and also for the collaborations, and again, for the organizers.
Thank you so much. That's a very beautiful study and also like very useful characterization of different tools, so people in field can like more critically assess alpha-min sense if they are going to use it. And I see there is a question from Yamina, or that at least Yamina's hand is up. Yeah.
Oh, my bad. Sorry. It was completely a mistake. I was trying to read the chat.
Okay. Is there a question from somebody else? If not, I will ask a question. So given that you built your pipeline on like comparing the things with AlphaSense, do you think if AlphaSense wouldn't exist, would you be able to like construct similar like model for variant prediction, or would be like do you think in that way is a bit biased towards what was available before?
Well, there are all other tools we could also use or analyze other tools as the starting point. But yeah, the principal idea that we had was to build one that was GPCR specific. But when we were about to start the project, alpha-min was published, and we saw that indeed it was a really good algorithm. Although the previous ones, as Eve or Rebel, although also had good performance, but none of them was especially trained with GPCRs. And we think that this could really impact the performance of the algorithm in order to be a specialist of GPCRs.
Cool. Thank you so much.
Thank you.
And just in 1 minute, we will like start with lunch session. So I thought it's good moment that I give you a minute or two to refill your cups, like find your lunch or dinner or breakfast if you intend to have one. And in a moment or two, there will be Ian telling us how lunch session will be structured. Thank you to all presenters. It was a very nice session. And if somebody has questions for them, you can find them